Okay, good afternoon, everybody. Welcome you all to this SMCB Seminar Series. This is the 9th lecture in this seminar series, and it's a pleasure to have as a speaker today Dr. Biman Jana from IACS Kolkata, and the session will be chaired by Professor Sandeep Paul from IIT Guwahati. So before I request Professor Pal to initiate the proceedings, I'll just mention a few things that in this talk we are going to record the talk, and also since Dr. Jana has agreed, we are going to put the talk somewhere in the YouTube. So in future, whenever the speaker agrees, we'll continue to put the lectures online so that people can listen to them in future. Yeah.
Shumon,
Yes.
tell that this is the 9th.
Yeah, this is the 9th lecture in this series. Yeah.
That's very nice because next one will be 10th.
Right.
That's a very strong arithmetic, but I think 10th is a good number. Okay.
Sure.
9th is of course a very good number according to Hindu. They tell you to buy apartments or houses with number 9.
Yeah.
Okay.
Yeah.
Okay,
Yeah. This series has been going on quite strong, and a lot of people have shown a lot of interest. Particularly students, both undergraduate and postgraduate students, have shown lots of interest, and I hope that this will continue. And so, as usual, we'll be taking lots of questions, and it is the customary to have a lot of discussion in this seminar. So I encourage all of you to please put your questions in the chat box. And we'll prefer to take the questions after the talk, but if there are any pressing question, you can raise your hand and ask during the talk also. So with that brief introduction, let me invite Professor Sandeep Pal to please initiate the proceedings. Thank you.
Yeah, thank you, Suman. Yeah. So good afternoon all. Welcome to SMCB Seminar Series. So today's speaker is Professor Biman Jana of IACS Kolkata. Biman received his BSc degree in 2003 in chemistry from Calcutta University, India, and MSc from IIT Kanpur in the year 2005. Then he moved to ISC Bangalore and joined Professor Biman Bagchi's group at the SSCU unit. During his PhD program, he has conducted research on dynamics of complex systems, bulk water, like hydration layer, thermotropic liquid crystals, conformational fluctuations in enzymes, using computer simulation and theoretical analysis. After a postdoctoral stay with Professor Jose Onuchik at UCSD and Rice University, he started his independent career at the IACS Kolkata as an assistant professor. Since 2017, he has been an associate professor and currently a full professor in the School of Chemical Sciences, IACS. His group is interested in theoretical and computational biophysical chemistry with special emphasis on exploring the mechanochemical cycle of molecular motor proteins and ice growth modulation by ice binding proteins. As mentioned, he worked on a large number of subjects from liquid crystal to protein hydration layer to enzyme kinetics and complex biological processes. He's quite dynamic and a highly active person. Moreover, he has also received the INSA Young Scientist Medal in 2018 for his contribution to the field of antifreeze proteins and motor proteins. So I welcome Biman to start his talk. Yeah.
Thank you, Sandeep, for a nice introduction, and thank you, SMCB, for giving me this opportunity to talk. Let me share my screen first. Okay. Is it visible now?
Yeah, it's visible.
Okay. Okay, so let's start. So what I'll be talking today is about ice binding protein, as the title suggests, and the modulation mechanism for ice formation in biological systems. Okay, so before that, I would just like to say that in my group, we use statistical mechanical theory and mathematical and computational modeling of biological systems. We work on motor proteins. We also work on ice binding proteins and work on phenomena like protein folding, aggregation, little bit of allosteric regulations, and also redox oscillator models kind of system. So today, as I said, I'll be talking about the ice binding proteins. But before starting to go to ice, let's start with water. Okay, so we all know water is apparently very simple molecule. It has one oxygen and two hydrogen having a V-shaped kind of structure. That's the molecular structure of water. But in ice, they form extended hydrogen bond network, and one water molecule can form hydrogen bond with other four water molecules, giving a perfect tetrahedral structure. In liquid water also, there are extensive hydrogen bonding between water molecules, but this is not perfectly tetrahedral. It is dynamic and it fluctuates. Few things about water and few really interesting things. First one, we all know that when it goes to ice, the density actually decreases for all, mostly for typical liquids. When it goes from liquid to the solid state, density actually increases. But for water to ice transition, ice actually, there is a decrease in density, and that's how ice actually floats over water. Then if you really see the density dependence, temperature dependence of density of water, it actually goes through a maxima around 4 degrees centigrade. And then if you think about the thermodynamic response functions like isothermal compressibility, thermal expansion coefficient alpha, kappa T, or Cp, which is our heat capacity at constant pressure, what happens is that for typical liquids, if you decrease the temperature, those things actually decreases monotonically. And as the third law of thermodynamics suggests, it will go to zero, monotonically decreasing to zero. But for water, you find really a different behavior. It actually decreases initially, but then for kappa T and Cp, it goes through a minima and then goes up. This is really very different from typical liquids. And for isothermal expansion coefficients actually becomes negative after 4 degrees centigrades. So water, even if it has a very simple molecular structure because of its extended hydrogen bond network and other things, they give really different and unique behavior. If you want to really read more about this and also biological consequences, structure, function, then you can read this book by Professor Biman Jana, Water in Biological and Chemical Systems, Chemical Processes by Cambridge University Press. Okay, let's now think about ice to water transition. First, phase diagram. You have liquid water, then you have ice, but ice has many different phases. What we'll be doing today, naturally abundant ice is ice 1H or IH, that is called hexagonal ice, this kind of transitions. So again, few things to know. If you take a glass of water and put it down, what is interesting, we all know that it has a 0 degrees centigrade, that is the temperature where it goes to ice. But generally, if you put it in a cold environment and decrease the temperature slowly, you will find even below 0 degrees centigrade, if you do not disturb, it still stays in liquid state. But once you disturb, like it is shown in the figure of the video, it actually forms completely ice. So that means you can really keep water below its melting points. And this kind of situation is called supercooled situation. The liquid staying liquid below its melting or freezing temperature. Okay, one thing is that. Other thing, if you see the density across this phase transition, there is a sharp change in density. Okay, another important stuff. If you think about entropy, you'll see that entropy suddenly changes in this. If you decrease the temperature, this is the melting or freezing point where entropy has a sudden decrease. For freezing and melting, there will be increase. And also, most importantly, this transition is associated with what is called latent heat. That means if you decrease the liquid water temperature, it will come to 0 degree. Even if you take out heat from this, liquid stays, temperature stays there, temperature doesn't change as long as the liquid completely converted to solid ice. So point is that all these things I have mentioned, this is actually trademark of a first order phase transition. Now, for first order phase transition, if we think about the theoretical perspective, that Landau came up with this theory of first order phase transition. What it tells you that you write down a free energy functional in terms of order parameter m. Now, what is order parameter? Order parameter is a quantity which differentiates between the parent phase, in our case water, and which is and the daughter phase, or we call it ice here. And generally speaking, m is 0 at parent phase and non-zero as ice phase. Okay. Now, this free energy functional can then be expanded in terms of Taylor series around the parent phase, or m equal to 0 water phase. And this is the expression for first order phase transition, where alpha t has this temperature dependence, b and c does not have explicit temperature dependence. But what is interesting, if you plot this function keeping some of the constants, that alpha or c equal to 1 and b equal to, let's say, -1, what you find, there exists a temperature called Tc, where if you see the middle plot, where you will see it has this kind of variation, that is a minima at m equal to 0, which is water, at a non-zero like 0.7 here, that is a minima called ice minima, and then there is a barrier in between. So that is interesting. And if you go to little bit above Tc, you also get these two minima with a barrier here. But if you go, this Tc is in our case the transition temperature, or what you can call the melting temperature. Below melting temperature, you have, of course, ice phase is stable, then the liquid phase, but still there exists a barrier. Now, if you understand the point I discussed earlier, so if you cool down a liquid, even if at T equal to Tc, of course, T greater than Tc, water is stable. T equal to Tc, two phases have the same free energy, but the problem is that because of this barrier, it cannot go to that other state. And that's how it stays in the water state. Even if you go below T equal to Tc, the same situations happen. Water try to stay in the liquid phase itself, even the ice phase is stable because of this barrier. And this kind of situation is called the metastability or supercooled kind of situations. Just to mention that this temperature T0 here sitting, I mean, you can see that all this temperature I'm talking about, which is T minus T0 equal to 0.20. So T0 is just a little bit lower temperature. The significance is that if you go to T equal to T0, between the two phases, this barrier actually vanished. And then water spontaneously goes to ice. And this kind of temperature is called homogeneous nucleation temperature. And for ice, it is minus 39 degrees centigrade. That means you can keep water theoretically as water even up to minus 39 degrees centigrade. Okay. Now, the point I wanted to mention here is that, so how does really it happens? Let's say if you take supercooled water, there will be density fluctuations in your systems. Some rare density fluctuations will form ice embryo in your system, but if it is too small, then it actually goes back again to water. But with rare fluctuation, at some point there will be critical nucleus formation, and then growth will take over the whole system. So to understand what is really happening, classical nucleation theory actually tells us something, that how to model this system. So you have this parent liquid supercooled, and the formation of the ice embryo inside this parent liquid. So then what is really happening? You are going for some water molecule, you are going to the ice state. Of course, because it is supercooled, as I told you, that means below Tm, free energy wise, there is a favorable term because you are forming the stable phase, and that is will be 4/3 pi r cube volume into, this is the free energy gain by changing unit volume to the ice. Okay. But there will be an unfavorable term, as you can understand, because of the formation of the surface, because ice and water is very different. So that means there will be a surface. So there will be a term which is your surface energy term. So that it will be a combination of the two. And it can be shown that at low radius, surface energy term wins. At high radius, this volume free energy term wins, and thereby it keeps a kind of goes to a maxima. So at the maxima, you can see what is the n star. That means the critical cluster size. This is this kind of expression. Delta G also have this kind of expression. So that means what I'm trying to say, up to certain point, up to critical nucleus, this is the nucleus formation. Once it is done, it will go to the growth phase, and there will be no blocking to the formation of ice. Okay, this is kind of, you take pure liquid and this is what happens. I'll just tell you another way of doing it, that is called the heterogeneous ice nucleation. That means what happens here, that ice actually forms over a surface. Okay, when it forms over a surface, then what happens, that this is the situation. That means ice grow on the surface, and there will be concept of contact angle will come, as you know, from a solid surface if something else is going on. So I'll not go into the detail, but if you do this simple geometrical calculations and do the same kind of theory you have done for the homogeneous nucleation theory, classical nucleation theory, you will get this barrier, as I told you, that will be a homogeneous nucleation barrier with a component shear, which is a function of contact angle. Now, what is that? If the contact angle is 0, as you can see, the barrier will be 0, because simply if you put 0 here, you can do this mathematics, you will get the barrier 0. But when it is 180 degree, you will see this becomes 1. So that means it will be like homogeneous nucleation. That and contact angle actually will be between 0 and 1. So that means your delta G of heterogeneous nucleation will always be less than homogeneous nucleation. Okay, and in our biological cell and other surfaces, you actually see this heterogeneous nucleation, not the homogeneous one. Okay, so now this, what I'm trying to say, that this freezing property goes through a freezing point. So how you can really change this? One way to change is the called depression of freezing point, which goes through a colligative property. How does it work? If you have, if you put something in your water, depending on how much the solute you have put in that, it will change the delta TF, that is the change in your freezing point or depression, will be this equation minus Kf, Mb and Pk. For water is 1.86. And M is the molarity of the solute. It does not depend on the nature of the solute. And P is dependent on if it is a salt, then how many components you are getting, all this stuff. How to think about it? If you put something in water, then it is tendency of water go to the vapor phase decreases. That means if you do this phase diagram, then in pure liquid, if this is the solid line, if you get for solution, the vapor pressure actually decreases. That means it will be something like this dashed line. So you can see that this is the point where probably initially for pure water was the freezing point. Now it is shifted to this little bit lower temperature. So this is colligative property, simple way to think. Today, what we are trying to think about is something a little bit different. That in biological systems, there are proteins which can actually change this process by specifically interacting with ice. Okay, there can be two ways it can be done. One, they actually interact with ice surface to enhance the process of nucleation. We call it ice nucleating protein. And there are proteins which actually prevents the growth process through specific interactions, and we call those antifreeze proteins. Okay, first we'll start with antifreeze proteins. So let's start with that. So what are those antifreeze proteins? The idea is that if you go to a peak or very cold conditions, the temperature is so low, your body fluid will actually freeze and become ice. And as I told you, ice formation, the density is lower. So your cell bursts. So organism cannot really survive. But in Arctic condition, there are organisms that do survive. How do they do that? They actually express a specific kind of proteins. It's listed here. There are many more. So that protein actually somehow prevent the growth of the ice by binding to the growing ice surface, and thereby they keep the body fluid still liquid. Okay. we'll first try to understand that how do they do it, okay, from a more from a more theoretical perspective. Okay, so let's say how it works. Let's say this is the growing ice surface which is growing. You have in cellular fluid, you have different proteins and you have antifreeze proteins. And I told you antifreeze proteins can bind to the ice surface. So they bind to the growing ice surface. Wherever they will find, there will be bind, there will be no growth, but there will be growth in between. That means you are forming a curvature growth, and that will have radius which is smaller than the initial one, right? And this smaller curvature growth has some implication, because if you see, I told you there is a critical nucleation beyond which the growth starts. Now, if you put a lot of AP on the growing ice surface, your R will decrease, and once it will decrease below your RC, then there will be no growth. And this kind of effect is called Kelvin-Thompson effect. I'll not go into the detail. You can write down what should be the difference in delta G. And there is a formation of curvature. Excess pressure for the curved surface comes to sigma by R. R is the radius. The smaller and capital R should be taken as same, and sigma is your the interfacial or surface tension. Using this, what you get, there is a relation between R and this delta T. Delta T means how much you can supercool. You can see that keeping everything same, by depositing the antifreeze protein, if you can make this R go low, then delta T from this expression will be more and more. That means you can keep body fluid still liquid or supercool it much more. Okay, so this is the way antifreeze property works. Now, if this is the situation, one thing is for sure, it has to bind to the ice surface. So first thing I am trying to say that how do they bind? Okay, so there are theories or hypotheses how do they bind. The first one is comes with the crystal structure of a very simple antifreeze protein, which is straight helical structures. But what they found that this has threonine well spaced, and that matches with the surface, ice surface it binds. Of course, one will say, oh, threonine has wedge group, so that means it is binding to the ice surface by hydrogen bonding, right? But then when people mutate this threonine by valine, that means the OH group is now mutated to another CH3, they find it is still active. But when threonine is mutated with serine, that means threonine has another CH3 group that is now become just hydrogen, then it becomes inactive. This hypothesis got into trouble. Now, snug feed model came up. It says simply that ice surface is flat and many ice binding protein has flat surface. So that means they can just fit on that surface. Okay. Now, we started this problem with this type 1 antifreeze proteins. So I said there are threonine, and activity data says if threonine is taken by valine, that means this becomes 2CH3 active, but serine not active. We have done a lot of things, but I'll tell only the important thing. We did the binding study. How do we start? We take the ice surface. First, we take threonine 1, and we actually started with hydrogen bonding structure. That means threonine wedge are making hydrogen bonding with the ice surface. But when we run the PMF kind of binding calculations, what we find that is not the stable state. Stable state is something different. It rotates. CH3 now point towards the ice surface. OH got out of the way. And there is a formation of clathrate around that CH3 that actually interacts with the ice surface. Okay, as you can see here. For valine also, we find the similar situation. So that means it is not really the direct hydrogen bonding, but clathrate mediated hydrogen bonding, clathrate formation around the group. That clathrate actually makes hydrogen bonding with the ice surface. Okay, and what is really interesting is that if you see these two mutant which works actually, one is wild type, another is valine mutant, they actually in solution form this kind of clathrate structure. But for serine, we don't find any clathrate kind of formation because it lacks the CH3. Okay. Then we go into another one. This is a very different kind of protein. It has a threonine ladder. and this tuning ladder on the ice. And if you see the crystal structure, you get there are really 4 or 5 ordered because space we make hydrogen bonding with the ice surface kind of initial configuration, but it is not stable. Again, the stability comes with the layer of clatrate. That means CH3 is again facing the ice surface. There is a clatrate formation, and that clatrate actually interacts with the ice surface. Okay, and that clatrate becoming stable because of the anchoring effect of threonine groups. Okay, so now I'm telling clatrate, but I have to say that how can I really see their clatrate? So for that, we can take out the help of an order parameter called F4 order parameter. Nothing, it is just take out 2 order dimer and get the 2 farthest hydrogen. And then this is the dihedral angle you are talking about. And the F4 is just cos 3 theta. It says for clatrate, it is positive, like pure clatrate 0.7, F4 0 for bulk water, and minus 0.3 for ice. When it is bound, this is the black curve I'm talking about, you see it is skewed towards the positive side. That means they are clatrate. But if you decrease the temperature further, there is a transition from clatrate to ice for those water molecules. That is what I'm trying to say. It binds with the clatrate, but then with lowering temperature, those clatrate becomes part of the ice surface, and then this kind of protein gets attached to the ice surface. Okay. We have done all diverse kind of structural diversity of the antifreeze proteins, and what we find actually that it is a common universal mechanism. That means they come with anchored kind of clatrate structure, they adsorb on the ice surface, and then they attach finally with lowering temperatures. So this is a clatrate dependent ice binding mechanism. Okay, and recently this water, that clatrate, which is not like ice water when it is binding, has been actually proven by David Baker group with the collaboration with us using SFG study. Okay, so far clatrate ordering around the surface, it's a universal mechanism for different things, different antifreeze proteins, and after adsorption, water goes to ice. Then what happened that in the field, they found another crystal structure of antifreeze proteins, typical 4-helix bundle with lots of water in it. Good news is that those water are clatrate, and it is known that they actually help in binding. But something else is problematic in this structure. We'll see what it is. If you think about the protein folding theory or funnel model or whatever, only thing I want to say, the crucial step is this chain, which is unfolded state, they first collapse. Okay, and then water get expelled, and you have a hydrophobic collapse. What is the consequence? Consequence is everywhere in PDB. You will find there is a dry hydrophobic core, no water. You already know what I'm talking about now. Now, if you go back to this structure, there are lots of water in between. So then, is this protein is not following the protein folding concept, or is it a exception? What is the surprise here? Okay, for that, what we did is that first we studied the temperature dependence of this. Okay, the temperature dependence of this protein tells you something interesting. It tells it is actually stable with the structure of lots of water within the core at low temperature where it works. Okay, as an antifreeze protein. But at high temperature, this gets really unstable. This RMSD and SASA tells me that. Now, unstable, but what kind of structure? For that, we did replica exchange molecular dynamics study. What is that? You take replica of the same system at different temperature, you simulate, and then every now and then using Metropolis algorithm, you swap the structure between temperatures, and that's how you get a better description of different structures and free energy at a particular temperature. Okay, what you find at low temperature, it is the active state, lots of water in it, stable, but at high temperature, it is the collapse state, like protein folding, whatever it says. Like water gets expelled and they come close. And this yellow are actually alanine. This protein has 60% alanine. That tells, and most importantly, the water, we didn't realize that there are 2 layers of water. Okay. Then we said, okay, that is fine, but try to understand that what is really happening. So if this is a hydrophobic kind of environment, let's try to do temperature dependence hydrophobicity. How do we do it? We calculate temperature dependence potential of mean force. First, we start with 2-methane. As expected, we get contact minimum. That means 2-methanes are in contact with water around it. We get a first solvent separated minima. This is also very known. That means 2-methane with the water inside, 1 layer of water inside. But what is interesting, if we go really below in temperature, that means in low temperature, there is the appearance of second solvent separated minima. What is that? Very simple, 2-methane with 2 layers of water inside it. Okay. Then we said, okay, protein has bigger hydrophobe wall. So let's go to some model bigger hydrophobe, rectangular hydrophobe or rod-like hydrophobe. What you are finding, essentially the same thing. Only thing is that you can see there 2 minima at low temperature has very similar value in PMF. But here, as we increase the hydrophobe side, the second solvent separated minima in 2 layers of water is becoming more and more stable. So this tells us that the structure of antifreeze protein with 2 layers of water has to probably do something with this. But before that, why we are getting this second solvent separated minima? For that, you can use finite difference method to get decomposition into entropic and enthalpy. What it does, you calculate PMF at a temperature and calculate at a different temperature, which is close to that one, and then use this formula to get enthalpy and entropy term. Like we know, first one, contact minima is entropy. Yes, we got that. But for solvent separated minima, we found it is enthalpic, and not only that, enthalpic term becomes more and more prominent at low temperature. But enthalpic term can come as methane water kind of interaction or water-water interactions. We did further decomposition, and what we get is really interesting. It is the water structure, water component that is actually stabilizing this. Okay, that goes the energetics part. But then come back to the structure. It's a 4-helix bundle. So we now designed our hydrophobe alike this kind of protein. That means 4-helix kind of protein, 2 helices, and then change the distance between them and do the PMF. What we find is interesting. At high temperature, we find the collapse state is the stable. As you can see, this one is the collapse state. But if we go to low temperature, this state is no longer stable. It actually globally stabilizes to the second solvent separated minima with 2 layers of water. So that is can be called this structure as a weakening of hydrophobic interactions. Can we connect this to something else? Yes. There is something called cold denaturation. That means if you take a protein and go to really cold temperature, it denaturates. And people know this happens because of weakening of hydrogen bonding interactions with water getting inside the core of the protein. So what we are trying to say that this maxi protein, which is a typical protein with lots of water in the core, is actually topologically a cold denaturation kind of state, but it is active at that temperature. It is designed by nature in that way. Okay, this brings to fine. We now know what is going on in for this kind of protein, but then we thought that, okay, let's now think a little bit more about the water, because that is what I'm interested in. This water, 400 water molecules, we know why they are there. But most important thing is that they are like clathrate. Question is, what determines whether they will be clathrate or not? Okay, what a parameter of clathrate I have told, but I'll not tell all these things right now. I'll go to straight to this point and just to tell you that what are the factors making this water clathrate inside that protein. One, hydrophobic environment, like lots of alanine I said, that is very important. Plus the confinement, that is also very important. And other thing becomes really important. And you have really heard this term earlier, anchoring. So there is a hydrogen bonding from the backbone to the surrounding water molecule. This anchoring is very important for the clathrate water formations. Okay, so from here, if I tell you that till now what we learned, first part I'm not going to say. but it is clutch formation is important, but anchoring clutch formations. When there is threonine group kind of protein, I said that threonine does the anchoring job. But for helical protein, I said it is the backbone does the job. Okay. Now the question is, how does backbone actually can anchor this kind of situation at low temperature? To understand these questions, we actually first see structural aspects. For antifreeze protein, we have seen the backbones are exposed to the water. That means they are making more hydrogen bonds with the water. Yes, that's what we told earlier. But for standard helix, it is the more compact hydrogen bonding going inside the protein, inside the helix. It is not really exposed. Then we see the number of residue per turn for the antifreeze proteins. We find it is 3.7 residue, but for standard helix it is 3.6. It is suggesting something that for antifreeze proteins, this helix is a little bit swelled, and probably this little bit of swelling compromises the backbone hydrogen bonding little bit, and that is allowing the backbone water kind of hydrogen bonding. And probably that is the basis for anchoring and giving you the clathrid kind of structure. But let's try to connect a little bit more. So I said standard helix is 3.6, but that is in abundant condition. What happens if you put it in cold water? That means cold denaturation kind of simulations I want to do. So I take a standard helix mini protein, which has a helical part, and then try to do the temperature dependent study to see that at cold denaturation temperature, or when the cold denaturation temperature happen, cold denaturation happen, and what is the structural aspect of this helix. What we find is really interesting. In normal condition, as expected, it is 3.6 helix per turn standard helix that is stable. But around 230 Kelvin, there is a transition and helix swell to go to 3.7 residue per turn. Remember, that is what is the value we got when we are analyzing the helical antifreeze proteins. This tells me something is going on. So this cold denaturation gives you a swallowing helix. That's fine. What else it does? As you decrease in temperature, the swallowing makes, this plot says, the protein water hydrogen bond increases, nothing else. And that is expected, as I said, swallowing, compromising with the backbone hydrogen bond, and giving you the backbone water hydrogen bond becoming more and more. Okay. So now, this is the thermodynamics. It is telling you one thing I just want to say here, that this is the free energy. That means it says that the denatured state is getting more and more stable at cold denaturation, after cold denaturation. And we can do this kind of same kind of finite difference method and get out entropy and enthalpy, and it find it is negative enthalpy. So that means it's an exothermic process. And cold denaturation is actually exothermic. And free energy calculation says different kind of helix transition, nothing else. Okay. The question is that why it is happening at 230 K? The answer is already given by Professor Saito, Professor Bakshi, and Professor Omino in 2018 paper. He said that it is actually at the same temperature for bulk water, there is some interesting things goes on. What is that? One, density changes is fast at that temperature, and that is associated with a maxima in the response functions. Okay, and this kind of scenario maxima has been implicated to something called liquid-liquid transition. What is that? If you see the super cooled phase diagram of liquid, so there is the lowest, there is a called glassy system, glass transition happen here, and glass can have two phases. One is low density and high density, and they're separated by a first order phase transition. Hypothesis is that why it is no man's land, because after supercooled water, this is the homogeneous nucleation temperature. Here, water should not exist, right? So then what happened that this line can extend to this liquid state, giving rise to HDL and LDL, high density liquid, low density liquid kind of situations. Critical point, and there can be criticality beyond this critical point. We call it Widom line. I will not go into the detail. It says that around that temperature, actually it crosses this line and there is a transition from HDL to LDL that gives you this particular scenario. Okay. So that means this is related to cold denaturation is related to the liquid-liquid transition. Okay. So now point is that if it is so, then changing water model should change this helix transition simply because at a different model, you have different liquid-liquid transition. And that's what we find. For T4P, we say it is 230K, but for SPC, we know that liquid-liquid transition is around 185K. And this helix pattern shows transition around that liquid-liquid transition. So that means it has to do with water. Okay. Now point is that fine. We are all talking about here trip case protein, helix, how it is working. But what about the antifreeze proteins? They also go through the similar transitions. That means at high temperature, this becomes 3.6 kind of scenario, but then around 230K, there is a transition to go to the 3.7 kind of helix. Okay, so what we are trying to say here, taking all these things together. This is the cold denaturation. It is active here. And then at low temperatures goes to an inactive state by cold denaturation. For antifreeze proteins, it is actually inactive here, but as it has to work on low temperature, after the cold denaturation kind of transitions, the swallow and helix, I said 3.7, there it becomes active. Why? The swallow and helix gives you more and more backbone water kind of hydrogen bonding interactions, and that gets anchoring effect to the surrounding water, make the water clathrate-like, which is important for the binding to the ice surface. Okay. So this is what I am telling, that 3.7 helix can have anchoring effect, but 3.7 helix has reduced. Okay. Now, I'll tell you few things like a story. One, we know that different antifreeze proteins can bind to different ice surface. Now, who determines that, or what determines that? What we find is really interesting. I said it comes with anchored clathrate water. But depending on what antifreeze proteins you are studying, that anchored clathrate structure has slight changes. And what we find, let's say, for example, hyperactive insect AFP, what you find, this is the clathrate structure, kind of 6-membered, 5-membered fused. It actually interacts with the basalpine, which has a similar kind of synergy in structure. But if you go to type 3 AP, which binds to the pyramidal or prism plane, you find a different kind of clathrate formation, like 5-membered mostly, and that actually resembles the prism kind of plane structure of ice. So that means I'm telling that synergy between the clathrate structure, it brings it in with, and the structure of the ice surface actually determines the plane specificity of binding. Okay. Now, try to understand all these things I'm talking about is the one surface which can bind to ice. What the other surface of protein is doing? That means non-ice binding surface of the protein. The question is whether they're similar to other protein surface or not. Answer is no. There are differences in amino acid compositions, as I have shown here. But most important thing to take back from here, amino acid composition difference gives you something interesting. If you do a hydration study of temperature dependence, you will see non-IBS keeps water liquid-like or resists to freeze more than normal proteins. What is happening here? Ice binding surface can bind to ice surface. Non-ice binding surface keeps water from freezing. So this is like a Janus effect. That means two surfaces doing two different things. So AP actually uses this Janus effect to stop the ice growth. Now, what we have done, interestingly, is real growth study of the antifreeze proteins. And you will find amazingly in this video that without water, it just freezes. The protein, it freezes. With type 3AP antifreeze proteins, it actually halting the growth. For non-AP of similar kind, it is pushing back and ice is forming. The similar situations happens for another antifreeze proteins. Water just goes to ice almost immediately. For antifreeze protein, it just stays there bound firmly. But for non-antifreeze proteins, it just pushes up and ice go on forming. So point is that whenever it does that, there is a formation of curvature, as you can see from here, like from here to here. So that can be seen better way that when it is bound, there is a formation of curvature. So that means, as you can see, the curvature growth is actually important for operational Kelvin effect. Remember? So that means our Gibbs-Thomson effect. So we actually show that this is the mechanism and it is actually working here. Then we did little bit of analysis just to tell you that just when it binds, there is this plot tells you how much is the water ice-like and clathrid-like, depending on the F4 parameter, whether how much fraction is F4 has positive for water, how much fraction is negative. And it says similar when it is just bound, but after that, I just told you that it becomes a part of the ice for antifreeze proteins, and you can see ice-like water is increasing and clathrid-like water is decreasing for antifreeze proteins. But for non-antifreeze protein, as it does not bind continuously increasing, continuously it is getting expelled, it never changes. Okay. One important thing I want to say that, remember, growing ice surface, AFP binds curvature growth. But this binding has to be nearly irreversible. Why? Because if it is reversible, that means, let's say there is a curvature growth formation and then AFP goes out. Then of course ice can form there again. So that means you will not be able to stop the growth. So that means you need an environment or need a situation where AFP can bind really in a permanent way. Now, how it is happening? Why they are having an irreversible binding situations? For that, for antifreeze proteins, I'll talk about this later. For antifreeze proteins, we first find before binding what is the hydrogen protein water hydrogen bond lifetime. We find somewhere around 150 here. Okay. But when it is bound, that means, sorry, around 200. But when it is bound to the ice surface, the value goes to 600. So that means there is a really increase in the protein water hydrogen, but it's becoming really long-lived. Okay. Now we form a very interesting situation. We said, okay, I know non-AFP will not bind, but just freeze it in the simulation and do the ice growth. So we find ice is growing, and of course you have frozen it, so ice cannot grow here anymore. So ice growth becomes now halted, and there is a formation of curvature also. Now at this situation, what we did, we make protein unrestrained. Interesting part is that even if we unrestrain, now there is no more pushing, it is actually making the ice growth stop here. Problem is why? For that, in this situation, we find what is the hydrogen bond, protein water hydrogen bond strength or time scale, that is around 150. But in this situation, when there is a curvature growth because of the restraining of the protein, that actually increases up to 400. And then after that, it does not expel from the surface. That tells me what is happening. What is happening that when AFP binds, first binding, then because of this slow unbinding kinetic curvature grows. So that curvature is ice. So once those water becomes ice, then ice protein water interaction is getting replaced by ice protein interactions. Now, because of the rigidity of the ice, your hydrogen bond lifetime increases. And once it is increasing, there are many more, right? So that means even if you break one, others will be there. It's like a chelating effect, keeping it simply there. Okay. So this is all I wanted to say about the antifreeze proteins. Now we have started also working on the ice nucleating protein aspect, but not too much. So remember, ice nucleating protein is the other part. It actually enhances the process of ice nucleation through specific interactions. Where you will find in plants, there is a frozen ice formation, and then plants leaf get really damaged, and it is actually done by a bacteria which promotes this ice formation and then damaging the plant leaves and get nutrients out of it. And then upon biological investigation, people have found there is a protein which actually pretty big. Here I'm showing only the smaller part that can promote the ice nucleation process and thereby damaging the plant. We have just started ice nucleating protein stuff. We have a model of this kind of proteins, and we first wanted to see only the hydration dynamics. Okay, for that we need an order parameter to see, and the order parameter we choose is something called Q6. Is the hexagonal bond orientational order parameter. It's simple. You take a central water molecule. There are other water molecule which is distance cutoff. You take the neighbor, and then you define from the center, and then other two water molecule, this azimuthal angle and psi and phi. Then write down what is your spherical harmonics, and from there, using this expression, you can get the bond orientational order parameter. Main thing to remember, for liquid water value is pretty low, this is 0.4. For hexagonal ice, it is 0.6. For cubic ice, it is 0.8. Okay, let's do the hydration and compare and contrast with antifreeze proteins. Interesting part is that for INP, we get this kind of temperature dependence. Okay, at low temperature, it is populating around 0.6, and there is an interesting peak around 0.8. It's kind of hexagonal ice. I'll not say ice. It is the water molecule which has hexagonal kind of local environment, and this is cubic kind of local environment. But what is important, when we calculate this parameter for antifreeze proteins also, we get similar kind of behavior. For SBWFP or hyperactive AFP, we get exactly similar. For type 3 AFP, we get more or less similar behavior. So that is telling me something, that the physical principle of interaction with ice for ice nucleating protein and antifreeze proteins are probably very similar. Now the question is then why one is doing promotion and not the other. The question lies, this antifreeze protein's ice binding surface is pretty small. Okay, so that means probably they are not able to give the surface area or enough surface area to promote the formation of ice nucleations. But for INP, it is known it has a counterpart or extra part which can interact with the lipid bilayer. And nowadays it is coming into the picture. How do they do it? It actually aggregates many more INP using this lipid bilayer, and then many more INP giving you enough surface to form the critical nucleus of ice and promote the growth of the ice at a very low supercooling. Okay. So with this, I just will say what is our plans and what is the bigger picture of this kind of study in my lab. So you understand this is the process: liquid, nucleation, ice nucleation, and growth. So you see that antifreeze proteins, it does inhibit this process. Why it finds its use? So for ice cream, you really want it to be very smooth but cold. That means you don't want to form real big chunk of ice. Food industry, cryopreservation industry, where you have to freeze embryo, you have to freeze cell and other stuff. You want it very cold, but you don't want it to be ice. So that means a mimetic design for this kind of system will be really important. Second, promotion, ice nucleation, which we have just started. Where it will find its importance? Great problem is that global warming giving you ice melt at the Arctic. Situation is that things will get flooded. So can I prepare a mimetic which can control this process or promote the ice formation again? Most importantly, we can go to atmospheric problem. In our stratosphere, there is an ice cloud. That is very important. If it gets too thin, our environment gets warm. If it is too thick, sunlight cannot enter really efficiently, it gets cold. Our planet gets cold. Now, pollution disrupts this natural process, paint particle and everything. So a mimetic design really can restore these things. And of course, the fundamental aspects we'll try to explore the ice growth and nucleation and growth kind of scenario in confined systems like nano confinement, because they give beautiful ice structure and unexplored region or less exposed region of ice phase diagram. So that's all I wanted to say. And then acknowledgement, of course, this is the team. This is Sandipon, Sridip, Prasun, Rahul, and Saurabh. They are, these three are still doing the work in this field. They are now, he's a postdoc with Kendall, and he's a faculty at MIT University now. So now, BST-CRB, of course, I should thank for funding, and Cray Computing Facility at IACS for computing facility. And thank you all for your attention, and I'll be happy to really discuss about this.
Yeah, thank you, Biman, for your nice presentation. Okay, it was very good. Okay, so Arnab will ask few more questions. Okay, so Arnab, yeah, you can start.
Yeah. So I'm there. So yeah, so great work, Biman. And also this time I understood it a bit more than last time, where you had less time actually to talk about this. So I will start. I have a few questions. The first one starts with your video, where you are you are showing that by finger tapping the supercooled water, immediately the whole water becomes frozen, right? So my question is that there is a barrier, thermodynamic barrier, that is already present from the nucleation aspect. By finger tapping, how is it ensuring that there will be disturbances that will create a critical nucleus? It could be other way around also, no? So
no,
created, right?
Yeah, so by tapping it, you are actually taking the system out of the equilibrium, right? It's like a mechanical disturbance you are doing to the system. So any metastable state is really unstable in that kind of situation, right?
Okay, so it somehow has like randomly creates some kind of deviation from equilibrium.
Yes,
so that it creates an ice nucleus somewhere, and that propagates, right?
That propagates, yes.
So what is a typical barrier in this process? Typical barrier for the critical nucleation?
You mean the value of the barrier? That's a nice question, and I really don't have an answer.
Typical value. See, it must be more than KBT, but I wanted to know, like.
No, it is more than KBT. Not only more than KBT. I think it is somewhere around. So at 0 degree, I guess it is tens of KBTs actually.
Okay.
So that's why you can really, so as you know that if you decrease the temperature, that both the N star, that means the critical nucleus size, and barrier decreases, and you can actually cool it down to minus 40 degrees centigrade.
Okay. My second question is that typically we know that protein makes stronger hydrogen bond, and you have explained it actually later after I asked the question that why there are two layers of waters coming in between and making these interactions and all that. So, but is it then the strength of hydrogen bond that is the reason for waters coming in between the protein and ice surface, or is it something else?
Yeah, so see, the point is that the model study does not have any hydrogen bonding. It is just simply hydrophobes forming rod-like shape or something like this, right? And you find there two layers of water coming.
Right.
So I would say that it is the weakening of hydrophobicity is possible.
But that is your second example. But in your first example, when you talked about, I think, threonine group and ice.
Oh yeah. There of course it is hydrogen bond for sure. Yeah.
That is the but protein would make a stronger hydrogen bond typically, right? So instead of water, like water ice interactions, strength of hydrogen bond, is it more than protein ice interaction hydrogen bond? Like
no. So I said two things. One is that that kind of threonine kind of protein, when it is in solution, it actually interacts with those water molecules and form a local structure around it.
Okay.
That is a clathrate kind of structures.
Yes.
And then if you have growing ice surface in your system, then with that clathrate, it adsorbs on that ice surface.
Okay. 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 Fine. Thanks. Now, my other question you have already answered. So all proteins will go to 3.7 helix at a lower temperature, apparently, right?
I mean, helical protein, if it is going through the cold denaturations. And I think your question is then whether all protein will be antifreezing or not.
Something like that, yeah.
Yeah.
But then that also, I think that you answered that,
Yeah.
typically what will happen is it will not have that interaction.
Exactly.
Although it will open up, but it will not have it.
Yeah, I'm just talking about some aspects that are similar to the cold denaturation. Yes.
Now, last one is that, when you put one antifreeze protein in water, so there is some region in which the curvature will be formed. So therefore, it will have its own effect, some length scale that it can monitor. It can modulate, beyond which it will not, and ice should grow.
Yes, that I have seen, and I am not showing it here. We actually change this by surface area of the growing ice surface, and when you go beyond a certain ice surface,
then
it will just invert the whole protein.
I wanted to know, typically, if you can say right now, what is that length scale? Is a double of?
For our case, we find around 8 cross 8 layers of ice for one protein.
Okay. 8 cross 8 means typically 8 into 324.
Yeah, something like that you can say. Yeah, 2 nanometer or beyond that.
Okay, that makes sense. But then in counterpart of that, you showed that if you freeze a protein, restrain a non-AFP protein, that also creates the curvature. And
because you are restraining.
Yes. My question is that instead of AFP, if you put any random solute and restrain it, it should have the same effect.
Yes, it is just geometric. But the point is, any random solute will not make stronger protein water kind of hydrogen bond, so it will expel. But in this case, it is making stronger protein ice kind of curvature ice interactions with protein surface and curvature ice water molecule, right? That is making
both are important.
Hey, Shondeep.
Yes, sir. Yes.
I think I want to ask some question, because this is going on this kind of personal thing. Personal thing should be on the point through other people also involved. Now, so sorry to butt in, but it was I waited. Now, Biman, you did a very good job in the phase transition part and these things. Now, when you have such strongly interacting system, ice on protein or protein on water, particularly you are bringing an ordered phase in connection with a semi-ordered phase. And in this case, there is a very well-established language in physics we call commensurate and incommensurate transition. You might have heard it in SSU.
Yes.
There is a lot of discussions on this commensuration and incommensuration. I think it will help you if you look into that, because what you are telling, actually, yes, there are some proteins which are commensurate and some proteins which are incommensurate. Now, talking of the surface tension in these cases is little dicey, because it does not get the molecular aspects of interactions. So if you now look into the heterogeneous nucleation picture, so first is look into commensurate, incommensurate. Second thing, please leave the heterogeneous nucleation slide on. They're very important. Another important aspect is that there is this famous model of Temkin, solid on solid.
Solid on solid.
You might have heard me talking of that. That is a paper in a crystal growth book. That paper was published, and I think I have a paper on crystal growth in Journal of Chemical Sciences, maybe Bijoy Damodar or somebody like that. I don't know. Way back. Where that reference can be, get the Temkin papers reference. But where you can see that is John Wick's roughening transition, which essentially Temkin solid on solid SOS model. And there, what they do, they kind of build an icing model like a picture. They bring, okay. So they bring one column on another column. Just a second, let me get my light here. Will it help? I don't know. I hope you can see the. So what happened? There's a flat, and then you have columns on that, and some column go deep, and so there's a roughening transition.
Okay, I got it.
And that is one of the ways a solid surface can get disorganized. And also in your gamma surface tension,
yes,
that expression I think can be extended in your case by taking into account these intermolecular interactions.
yes.
I don't know whether John Weeks, because that's a very famous work along with other work John Weeks has done, but roughening transition, when it came out 1976 physical review or 77, first one was Weeks, or I'll send you that PRL. Second one was a long physical review. This is a wonderful work, which is, I think, quite and Temkin, quite relevant to you. Now, the surface tension concepts, which becomes dicey in this case, but in a general context, one can generalize this heterogeneous nucleation expression. Actually, when you were giving this talk, I tried to call Montu Santra, which is a brilliant, quiet person, to actually get into he was not in the talk. I wanted him to hear, but he unfortunately is in hospital now with dengue. So, but he did a lot of work. So these kind of intermolecular interactions that go in and can become an extra parameter in your heterogeneous nucleation theory.
Yeah, I think some works are already there, how the surface tension gets modified in this kind of situation, when the interactions between the solid surface and the ice forming, if matching or changes, not only contact angle, I'm telling in terms of intermolecular interactions. There are some works done by Valeria Molinero from University of Utah. They are
but Valeria will do only, Valeria, I know her very, very well because I overlapped with her in Caltech. Valeria will do only numerical work.
Yeah, I know.
I am telling you more
on the
introduce a parameter in the heterogeneous nucleation, which can take care of the commensuration, incommensuration, or specific type of interactions.
Okay.
Because you see, when something is growing from the beginning, from a small, in a very early stage, nucleation concept is dicey.
Yeah, that is true.
Nucleation concepts become useful when it has grown to some extent. So what it's telling that there's a lot of very beautiful work you can do if you try to build analytical models. My perennial recommendation to people that actually I gave talk recently, several talks, and I showed the proud triangle and experiments, models, and simulation. And I think you should really try to build some models here and analytical models. I know a little bit of Molinaro's work, but I'm sure that she will do only simulation.
Yeah, mostly she's doing simulation.
I have never seen her doing any, I know her for a very long time now, never seen her doing any analytical work. So, but one can get some cue from her. This is what I wanted to say, that these are beautiful systems, but if you can build some models here, then many of the questions, say some of the questions we are asking, they can be answered.
Yeah.
that, like, for example, when you put the finger in and it becomes ice nucleation, then the question is that, yes, it is perturbed from metastable state, as you said, which is correct. But question then would come, how does it get perturbed?
Yeah, that is, of course, something.
And that perturbed is then, you would say, okay, this is a macroscopic process. What is happening below is 10 orders of magnitude faster. And very well, it also heterogeneous nucleation is taking place.
Yes. When I'm,
and that is the way it's getting out of metastable state. But for these things, you need a little bit of more microscopic theory, which we have been lazy. Except I have not seen analytical work on these. We are supposed to do something with MON2, but has not moved very fast. So my recommendation is that look into this, look into John Weeks, look into Temkin, and do into commensuration, incommensuration. You might get a more tangible explanation of the large number of interesting results that you have got. Huh? Okay.
Okay, so we have a couple of questions from Snehasis. Snehasis?
Yes.
Yeah, you can ask.
Very good talk. And I have a couple of questions about the class rates you have talked. So when you are defining a class rate structures around the hydrophobic residues, so are you calculating the tetrahedrality of the water molecules, the interfacial water molecules, or how you are actually?
No, I mean, I haven't done too much of tetrahedral, but I know that tetrahedral order parameter will be high here. I mean, we have done for some systems for sure, but the point is that what we find that, I mean, initially we started doing Q6, okay? Then we find that hexagonal order parameter is actually increases both for ice and both for class rate. I mean, there are extent of increase is different, but then we thought that, okay, then this is not going to work. I mean, whether we can see they're perfectly like class rate, not like ice. Then we went on to see that there are different other order parameters which can actually differentiate these two kind of phases. And F4 is 4-point order parameter is something which can distinguish between two states very efficiently. Yes.
Okay, but why I'm asking these questions, because when you in your hydrophobic solutes work, we have also followed some of your works. There, the ordering of water molecules around the hydrophobic solutes is pretty high, and that we can understand. But near the amino acids, which are like some hydrophilic groups are also there nearby. So it is often said that the presence of these hydrophilic groups somehow disrupts these class rate structures, which should be there around the hydrophobic residue.
Yes, I mean.
So I was actually was thinking that whether this ordering is really there around these hydrophobic residues, or some other parameters you have calculated which.
No, ordering will be there. It's the point I'm trying to make here is that around hydrophobic solute, there are ordering, there is no question about it. But the point here that threonine is a residue, if you see that it is everywhere in the ice binding proteins, simply because of the fact it has dual character. One CH3 group that can give you a hydrophobic environment to form this cage, and then OH group can actually anchor it. This is a very special group. That's what I'm trying to say.
Okay. Another question I had. So this is a very beautiful explanation that this cold denaturation of protein is because of this water liquid transition. And I think previously the explanation was like hydrophobic interaction decreases. Somebody says
I mean, hydrophobic interaction decreases. I have no problem in that.
Although it was seen only for this simple liquids. Yeah. So the point is this liquid transition. If that is the only reason, then do you think that all protein will have the same cold denaturation temperature? But in reality,
no. See, the point is that something is, see, you will see. I mean, I did not get time for this strip cage and antifreeze proteins. They are showing similar transitions, but transition temperature is little different. The bulk transition happen 230. Depending on your specific protein topology, the surface transition will be different. That we have shown in some other works with Professor Bakchi at the time of PhD, that for DNA, different groups like major group, minor groups will have different transition temperature actually. So that specificity will actually tell you that which one will show at what temperature. Yes.
Okay. Okay. Very good. So last questions I had. So if you see that whatever model you have taken, like T4P 2005 model, and from Professor Bakchi's work, it was like 230 Kelvin temperature was the transition temperature. But my concern was like when you were seeing this, that was in bulk water, right? Now when you are added this protein, so is this protein is not changing this phase transition temperature at all, or you are saying that
yes,
again the bulk
no. You will see that this protein is doing at 220. That's what I'm trying to say. It is not exactly at the bulk, but it is the effect of that. You can see that. That's what my point is. Yes.
Oh, okay. Fantastic. Man. Thank you.
Yeah, then we have a couple of questions from Somok.
Hi, can you hear me?
Yeah.
Hi, Biman, wonderful presentation and work. So I had two questions. The first one is like the initial part you showed that there are certain water molecules that are between two threonines in the threonine ladder in the AFP, and they form the anchoring for the whole clatrate structure. So do these water molecules retain their positions, or they can be
that's what the first thing I said. I probably a bit fast. I actually started the simulation to see that, to take crystal waters, even if they're there, I just strip that out. And then doing a simulation, it doesn't take even a nanosecond. Right. And then they are perfectly in that position. It's because both the sidewise, I mean, 2 threonine makes dual hydrogen bond with that, and keep it here.
So they cannot get exchanged with other water molecules.
Yes, it will be exchangeable, but the time scale will be pretty
right. Because I guess that would also affect the kinetics of binding, because the clatrate. Yeah. Okay.
You are right. Yes.
Yeah. Thank you. And the other question is that you talked about the swelling of the helix in AFP as compared to a normal helix. So can you comment on like whether there is a particular distribution of a particular kind of residues in the helix that allows this swelling?
Yeah, this is a very nice question. This is the first of a kind. I mean, I have to really look into this problem, both from the different helical composition and also in terms of dynamics also a little bit, because you know that I understand that both of them going to the cold denaturated state with 3.7 helix, but one is active, another is not active.
Yes.
Then what actually determines that? I think it will be, answer will be in dynamics little bit, and also the specific sequence little bit.
Because I mean, structure and everything is always determined by sequence.
You are completely right, but I haven't looked into it yet.
Okay. Thank you so much.
So the Devdas has a question. So Devdas, are you available?
Yeah. So I think I have two questions. One question actually I asked already, and I think you have already answered that question. The question was like whether the non-ice binding portion of the antifreeze protein, whether that can nucleate ice or not. I think
no, it cannot nucleate ice. It actually keeps water more like water.
Yeah, and the reason is I think the size is very small as compared to the ice nucleating protein.
Yes.
And that is why it cannot heterogeneously nucleate the ice.
You are right.
I got that from your last part of your presentation. The other question is a kind of like clarification question. So like when you mentioned that that peptide actually, you change the water model, like that transition temperature goes from 230 to 185. And so if I understood correctly, I think having Widem line doesn't mean that there is a liquid-liquid transition for a particular water model, right?
Okay, this is a different debate. I get your point. See, there are a lot more explanation between this kind of transition, that why this kind of density maxima and this kappa T alpha goes through this kind of peculiar shape. One of the explanation is through liquid-liquid transitions, and our earlier work with Professor Bakchi during my PhD, we showed that if we define the high density and low density liquid in terms of 5-membered, 4-membered, and 3-membered ring, our 3-membered, 5 coordinated, 4 coordinated, 3 coordinated water, we can actually see that the whole system is going from a low density to the high density kind of situations, which goes with the idea of having a liquid-liquid transition. Okay, I know that this is debatable whether the liquid-liquid transition is there or not. But I don't want to go into that right now, because liquid-liquid transition is something, still debatable. Yes.
Thank you so much. It's a very nice talk, actually. Thank you.
Yeah. So Navanita has a question. So Navanita, are you available? Or?
Yes, sir.
Yeah, you can ask. Yeah.
Okay, sir. Sir, I have a question, like at higher temperature or when you're changing the temperature, sir, is there any change in or denaturation in the structure of the maxi protein which you have observed?
Can you, if I go to the higher temperature, whether there is a change in the structure of the maxi protein? That's what we are trying to say.
Yes, sir.
Yeah, that's what. That it has 2 layers of water at low temperature, working temperature. If you increase the temperature, that water goes out and it goes to the model of hydrophobic collapse.
Okay.
Collapses.
Okay, and sir, one more question I have. In the Lauder theory of first order phase transition, so what is the T0 temperature like?
Yeah, that's what. If you go to T equal to T0, between the 2 phases, there was a barrier, right? That barrier vanishes.
Okay.
So water will spontaneously go to ice, and that is what is called the limit of stability.
Okay.
That is minus 39 degrees centigrade for water. Yeah.
Okay, sir. Thank you, sir. It's a nice.
Okay, since there is no more question, okay, so let us
I have one question actually.
One more.
If there are no more questions. So, so Biman, I was just curious that you said that when there is only one small hydrophobic group, then there was one layer of water or one layer. Like unless it become bigger,
no,
the 2 layers did not come into the picture.
2 layers does not become the global state.
Global state, right? No, global is still the contact minimum according to the PMF that you showed.
No.
Right. You showed later on.
Later on. That
so now I was just curious. Now, if I extend it further, if I make it even larger hydrophobic group, do you expect 3 layers, 4 layers?
I think that the theory calls it yes.
No, because beyond a certain point, beyond 3 layers, it will be just like bulk, right?
Yes, beyond 3 layers, I think it will not go. You might really get the 3rd minima if you really make it bigger and go. That's an interesting question, but it's
because the hydrophobic crossover will come after 1 nanometer size.
I know. So this is
where
already
is the saturation. Where is the saturation?
This is already 1 nanometer. That's my point.
Yes, exactly. That's why. Where is the saturation? Is it
you may not get another one actually.
Yeah. So that is the thing I was wondering. Okay.
Yeah. You're right.
Yeah, I also have one question to Biman.
Yeah, Rakesh, how are you?
Yeah, hello. Yeah, good. Yeah, so you attributed cold denaturation to the liquid-liquid transition in your computational studies. But as it is well known that in bulk water, we don't have liquid-liquid transition. So, and cold denaturation is also experimentally observed there. So what could be the possible reason there? I'm just wondering whether...
again, can you tell me what you were trying to say? That
but in bulk water case, we don't have liquid-liquid transition because we can't go to that, thermodynamic conditions. But cold denaturation is also observed experimentally.
No. If you want to really see the cold denaturation experimentally, you somehow have to stop the ice formation of your system. Otherwise, you have to go really to the cold temperature below 0 degrees centigrade for sure. Otherwise, so that's why you will find very little experiment in cold denaturation simply because ice crystallization actually interferes with that. There are specific experiments where they stop the ice formation.
One thing I think Rakesh's issue is that is for bulk water. Now, in constrained or confined water, I think there are evidence of liquid-liquid transition.
Liquid transition, yes.
Rakesh can correct me. Bulk water, yes. There are certainly a metastable minimum, but that it does not materialize because no man's land interferes.
No, yes.
But in a confined system, that what happens in bulk liquid might not be fully, because there are, I think, some amount of signature.
Yes.
So what Biman is talking probably far more confined systems, and actually
Yes,
that's a
I'm
very
talking-
interesting angle. That's a very interesting angle to be pursued.
Okay, yeah, all right. And the second question is that you are talking about the bursting of the cell, right, at lower. But when we think about in vivo systems, we have, first of all, we have effects of confinement. Second one, that let us say inside the cell you have 70% water but 30% external impurities. So external impurity as well as confinement is going to suppress the ice, nucleation as well as the growth.
Actually, it is.
Environment.
I know.
How important is the role of antifreeze protein for stopping ice nucleation at those conditions?
So Rakesh, the point is that as you are telling this, because of the presence of this kind of situation, the freezing point will depress. That's what you are trying to say, right?
Yeah, freezing point as well as like nucleation. Yeah,
but
right.
It is actually increases simply because in biological system it is more like a heterogeneous nucleation. Because of all different proteins and everything, it gives you the formation of ice well above the minus 39 degrees centigrade. You will get in minus 4-5 degrees centigrade. It will just freeze simply because of the heterogeneous ice nucleation comes into picture.
So okay, so it's like because confinement effect is also there, like very crowded.
Yeah, I know.
And
whatever happened, people have found
suppresses ice nucleation. So yeah.
Yes.
Okay. Yeah, but this could happen.
That suppresses ice nucleation, but heterogeneous ice nucleation picture actually elevates it from the real stability like minus 39 degrees centigrade. Overall, you get in pretty normal kind of minus 5 to minus 7 degrees centigrade, it will freeze. And antifreeze protein works on once the small embryo starts, it just stops the growth by.
Very so that you discussed. Yeah. Okay, good. Yeah, very nice talk, Biman. Yeah, thank you.
Okay, do you have any other question from anyone else? Okay, if not, then let us thank Biman for his wonderful talk. Yeah, so now I'm handing over to Sumon and Rajiv.
Thank you, Professor Shondip Pal, for kindly chairing this session. And I also thank Dr. Biman Jana for this nice talk. And we had lots of discussion, lots of insightful comments and suggestions has also come up. And so that's the whole purpose of this meeting. So I hope this continues. So before we conclude, there is one announcement. So we had discussed about different kinds of talk in our last seminar. So in addition to this monthly faculty talks, we had also discussed that we will have some intermediate seminar by senior PhD students or postdocs. So that is something was discussed in our previous seminar, but because of the vacation in between, puja and other things, we could not make it happen. So what we have decided that for the next iteration, next talk of this series, that will be on 4th December. What we'll do is we'll have 2 talks by a senior student or senior postdocs, and that will be only for this next seminar. And next time onwards, we may have this intermediate additional talks by students and postdocs. We'll continue to continue that activity. So please stay tuned, and we'll announce that who will be the speakers and the topics of the presentations by these senior students and postdocs. We'll let you know that in near future. So the next seminar will be on 4th December, and that will be by 2 either senior PhD students or postdocs. Okay. So we now have created a Google group. I'm sure that all of you are already part of it, and I'll encourage all of you to spread the news and maybe recommend your group members and friends who will be interested in this area of research. Please suggest them to join this Google group, and this is essentially a mailing list, and so people will be informed once for all of our activities that will happen in future. Okay, so again, I thank all of you for being here, and I hope this activity will continue as usual. So thank you all.
Thanks.
Thank you.
Thank you. Okay. Bye.
Thank you.
Bye, everybody. Yeah. Bye.
Bye.